Yes, start. Anupama garu, start చేయండి.
Okay. Good morning, one and all. Welcome to the second day session of HUL FDP conducted by IQAC cell of KVSR Siddhartha College of Pharmaceutical Sciences. I, Dr. B. Anupama, Associate Professor, HOD of Pharmaceutical Chemistry Department. It is my great pleasure to introduce my guru, Professor K.P.R. Chowdary Garu. Is one of the senior most professors of pharmacy in India, having 44 years of experience in teaching, research and administration in pharmacy. Sir was the founder principal of our esteemed institution, KVSR Siddhartha College of Pharmaceutical Sciences. He served as lecturer, reader, professor, HOD and principal of Andhra University College of Pharmaceutical Sciences, Visakhapatnam during 1977 to 2010. Sir served as chairman BOS in pharmacy in JNTUK Kakinada during 2010 to 2017. He is research director of IQAC Institute of Pharmaceutical Sciences and academic advisor and researcher of our KVSS COPS. Sir is the member of ACT South Regional Committee Chennai and also South Central Regional Committee Hyderabad. He is the expert member of NBA and NAC peer review committees for the purpose of the accreditation. Sir served as PCI inspector since 1980. He is the chairman and member of Board of Studies of Pharmacy of several universities like Acharya Nagarjuna University, Andhra University, Berhampur University, Padmavati Mahila University, JNTU Hyderabad and JNTU Kakinada. Sir is the member of the B.Pharm Syllabus Committee constituted by PCI. Sir is the co-convener of IPCA committee during the last 10 years. He is also advisor committee for NBA and NAC accreditations during the last 10 years. Sir also has industrial experience as a research consultant, TTK Pharma Limited, Chennai for 5 years from 1981 to 1985. He has research advisor for ESI Pharma Technology, Visakhapatnam for 2011 to 2014. Sir has published 446 research publications in various reputed journals and also total citations of 3022 and also I-index 67 and G-index 44. Sir has published 2 books for pharmacy students. One is Research Methodologies and Biostatics and also Pharmaceutical Quality Assurance. Sir has presented research papers 72 in various conferences and seminars and also delivered lecture as a resource persons in various seminars and conferences. Sir has research projects, handled 4 research projects and also patents. He has 5 patents and also he has guided 100 number of PhD students and also MPharm thesis and MPhil thesis 120 and also served so many awards, national awards and state awards. Among that, the Eminent Pharmacist 2019 from IPA Mumbai and also Lifetime Achievement Award from Biotechnology and Pharmacy India and also IPA Fellowship Award in 2009 from Indian Pharmaceutical Association and also as a President IPCA Rajahmundry Local Branch received Best Local Branch Award from times 14, 15, 17, 18 and IPA Mumbai. Sir also Best Teacher Award 2017 from Government of Andhra Pradesh and also Best Researcher Award in 2000 from Andhra University. So thank you, sir. Welcome to this session and thank you for accepting our invitation. Hearty welcome to the session, sir. Today, sir, going to deliver a topic on in biopharmaceutics and clinical roles, sir. Sir, proceed the session, sir.
Yes, shall I start, Madam Anupama, Madam?
Yes, sir.
My voice is clear?
Yes, sir.
Okay,
Yes,
okay,
sir.
audible. Okay. Thank you, Dr. Anupama, introduction and coming to our program. First of all, I congratulate Deval Rao, Principal of the college, and Dr. Vijay Kumar, HOD of Pharmacy Practice Department, as well as Organizing Secretary of this program, and all other faculty members who are acting as the coordinators. And I am very happy to associate with this function. And Anupama, she has the distinction here. She belongs to the first batch of B.Pharmacy students in the Siddhartha College of Pharmacy. I am very glad that she has later on excelled in the medicinal chemistry area, and now she is heading the Department of Medicinal Chemistry or Pharmaceutical Chemistry in this college and taking the lead in organizing teaching and research in this subject. And she has given a beautiful introduction. And just I want to add one more aspect that several of my students are very successful. Notable among them are in your college itself, Dr. Deval Rao, Dr. Buchinaidu, Dr. Ravi Shankar, and in all the neighboring colleges, all the principals of my students only. And moreover, yesterday, our chief guest, Dr. T.V. Narayana, happens to be my student in B.M.Pharm as well as PhD degrees. And hence, I feel very proud to say that my students excelled in their own way in different areas, particularly in academics. And I am very happy over this. And I consider it as my privilege to be the teacher for all the successful pharmacy graduates. And now Dr. Vijay Kumar, a few days back, asked me, sir, we are going to conduct a 1 week long faculty development program for the purpose of teachers and students also can join. And so he asked me to speak on a topic. Then I thought it would be better to discuss something about the clinical pharmacokinetics and its role in clinical pharmacy. Because clinical pharmacy, as all of you know, is an upcoming in India, though it is well developed elsewhere. In India, with the introduction of the pharmacy practice or PharmaD program in the country, somewhere in 2007, and it's getting slowly establishing its image in India also, clinical pharmacy, and particularly clinical pharmacokinetics is again a very essential component of clinical pharmacy, which will be very much useful to the practicing pharmacists as well as the future pharmacists, that is the teachers and students as well. And hence I have selected this clinical pharmacokinetics and its role, how important it is in the clinical pharmacy area. That is, the objective of this presentation is to highlight various aspects of clinical pharmacy, particularly pharmacokinetics, how it will be useful for the clinical pharmacist, and with a few case studies, etc. I have drafted this entire program. And this program, apart from learning the basic concepts, and later on, it involves a number of practice cases I gathered, and I will be discussing. And what is needed is all those listeners should pay good attention to the topic. Otherwise, we will miss the link somewhere, because there may be many mathematical expressions and numerical values, and hence normally it may not be very attractive. Even then, thinking the importance of the topic, I thought in a brief way I can discuss some of these useful aspects in this subject. And now, and hence this is titled like this. And then objective says so to highlight the principles of pharmacokinetics, clinical pharmacy, pharmacokinetic models and parameters, their application particularly apart from other things in the design of dosage and dosage regimens in normal subjects as well as the patients. And in the normal subjects, it is a major concern of the pharmacokinetics. And when this is applied to patients, and there will be a lot of difference in the patients and the normal subjects related to kinetics, with the result, the clinical pharmacokinetics, the emphasis is made on the kinetic behavior of the drug in the patient. And hence these aspects are covered along with a few case studies. Now, the subject is overall, the subject is known as biopharmaceutics, which include the pharmacokinetics. And hence, you cannot, there is no separation exactly between biopharmaceutics and pharmacokinetics. They go together anywhere. And both concerned with the performance of the drug and drug product in the body. That is the bio term refers to that. And then pharmaceutics means the dosage form. So what sort of relationship exists between the dosage form factors means the drug and its properties, dosage form and its properties, and the biological effect or pharmacological effect more particularly, and also to some extent the toxicological effect produced by the drug after its administration, and what sort of relationship is there between these two things is otherwise the subject matter of biopharmaceutics and pharmacokinetics. In the biopharmaceutics, we come across with the a study of the general principles governing the absorption, distribution, metabolism, and excretion. That means ADME. So the principles governing these will be very much useful in the drug design process, in the development of delivery systems, in the usage of drugs and drug products, etc. And hence, the pharmacokinetic part is also much similar because it also concerns with ADME only, but the kinetic, that means what is the time course with respect to time after administration, what is happening to this drug in the body, how the body is handling this drug, and you know very well that after its administration, the body will consider this as a foreign entity and will not allow to stay in the body and will try to eliminate it by different means. And hence, whatever you administer, the drug, it will not permanently stay in the body. If it stays permanently, it will be toxic. And certainly, it has to be eliminated from the body, removed from the body. And during this period, before it is eliminated, we have to derive the therapeutic effects of the drug, minimizing the possible toxic effects. That is the idea of the study in the pharmacokinetics. And hence, simply the pharmacokinetics, it concerns with the study of the rate kinetics of ADME and how these will influence the performance of the drug and the dosage form, particularly in terms of the therapeutic effects and toxic effects. And hence, the pharmacodynamic, there is another word which concerned with the relationship between the response means the effect and the concentration of the drug in the body means in the plasma and tissues. How these two are related, it is otherwise known as the pharmacodynamics. So it is also an integral part in the pharmacokinetics. Like this, you have this one important subject, biopharmaceutics and pharmacokinetics, which is considered as most important for researchers, particularly in those who are involved in the drug discovery development and including at all stages, the principles of biopharmaceutics pharmacokinetics are very much useful. We'll see a bit later some of the important things. And then the pharmacokinetic models that frequently we will be using. So model is, this is a mathematical expression which is used to describe, to characterize the variable. The variable in this subject is the plasma concentration, which varies because of the process of ADME, and it will not be stationary. It is continuously moving, and hence it's a kinetic model means the mathematical expression which is used to describe the time course of the drug concentration in the body, and basing upon the drug concentration only, we can achieve the desired effect or the toxic effect. And hence, the pharmacokinetic models, mathematical models, they are in a way providing a concise means of expressing the mathematical relationship associated with the concentration of the drug in the body. And hence, the pharmacokinetics majorly concerned with the mathematical description of the process of absorption, distribution, elimination, and excretion. Now, coming to clinical pharmacy. this background, the clinical pharmacy, you know very well, is I told you it's upcoming, yes, but it's well established in other areas, and it concerns majorly with the science and practice of rational use of medicines. Medicines are to be used appropriately, then only they serve the therapeutic purpose. Otherwise, they will be more dangerous, and hence they should be properly used. What is rational use is the right medicine, right time, right dose when it is administered to get relief from the disease or symptom. It is said to be a rational usage. So at every stage, there should be some rationality for its usage, right from the selection of the drug, right from selection of the route of administration, dosage regimen, and care, what care you have to exercise in using that, which depends upon the possible side effects of the drug, etc. All these are very essential in the practice, clinical practice. And hence the clinical pharmacy concerns like this, the scientific concepts involved, as well as the good practices followed for the rational use of medicines. And in this rational use of medicines, the main goal, what is the goal of this clinical pharmacy is to promote correct and appropriate use of medicines. Correct and appropriate use. And it involves with the objective of maximizing the clinical effect. Always you should derive the maximum possible effect of the drug and minimize the risk of the toxicity, as well as the drug-induced adverse effects are to be reduced or minimized, and minimizing the expenditure also is one of the important. That means in the rational use, it also includes the concept of cost, and it should be cost effective and should be reachable to all sectors in the people in the society, and hence cost also forms an important consideration for the pharmacist, and to deliver the medication to the patient in the reasonably affordable cost does not mean the low cost, and minimizing the expenditure is essential in this practice. Like these are some of the goals, and then you have several problems when you are using drugs, and normally drug related or medication related problems will arise. That means improper drug selection, right? The selection itself, the drug is not properly selected, and subtherapeutic dose, that is dose less than the minimum quantity required, and medication failure to receive, that means failure to receive the medication properly by the patient, and medication overdose, sometimes dose may be high also, and adverse drug reactions are possible always, and drug interactions, food interactions are possible, and medication use without indication. No, today, the drugs, you know that OTC drugs, there is no need for any prescription, and hence they are freely available. With the result, the patients or the public generally tend to use these medicines on their own. They're supposed they can, but with care only. But this is leading to the usage of drugs without any indication. There is no proper indication for the drug. Even then, the patient may be using. That's really a big problem for the pharmacist. And hence, all these are some of these problems can certainly be solved if the pharmacist possesses a good knowledge of pharmacy practice or clinical pharmacy. And the clinical pharmacokinetics is also very much helpful in resolving some or many of these drug-related problems in the practice. Now, clinical pharmacokinetics, how it is different from pharmacokinetics, you already see it, but this is the application of pharmacokinetic principles to the safe and effective therapeutic management of drugs in individual patients is the main concern in the clinical pharmacokinetics. And hence, patient-wise, he is suffering with a disease, and disease-wise, there is a possibility that the kinetic behavior of the drug may little alter, and hence the dose may or may not be adequate. Many times the doses have to be rescheduled, and basing upon the pharmacokinetics of the drug in the patient, and hence the pharmacokinetics of the drug in the patient are something different from the pharmacokinetics in the normal subject. And in the therapeutic books and in the literature, you find the kinetic parameters as well as the dosage, they are all worked out in the case of the normal population only, large number of normal population, and hence these parameters may not exactly fit into a particular case or a particular patient, and hence the clinical pharmacy should have a knowledge about how these parameters are likely to be changed in a particular patient, and you should be able to determine them quickly and use this knowledge or this parameter, revised parameter in his schedules, dosage schedules or other care. And hence the clinical pharmacokinetics in that perspective is a very essential concept and science of subject for the clinical pharmacist. And the objective being enhancing the efficacy and decreasing the toxicity of the patient's drug therapy. Whatever is the therapy, the possibility for the toxicity will be there, that you have to try to decrease it or minimize and get the largest benefit, enhance the benefit. That should be the objective. And plasma concentrations are affected by the pharmacokinetics. What pharmacokinetics will greatly influence the plasma drug concentration, or in general, the drug concentration in the body is the rate at which the drug is administered. The rate at which the drug is administered. It includes both the consideration of the route of administration, the dose of the administration, type of the dosage form. All these influence the rate at which the drug is entering into the body, and then the volume in which it is distributed. That is, the administered drug will not be there only in the plasma or any one particular tissue, but it will be rapidly distributed to all other tissues in the body or organs in the body, and hence the concept of distribution is also very important. And then clearance. All the administered drugs will ultimately be removed from the body, and hence clearance is the term used to designate the process of removal of the drug from the body. It majorly, you know, that it is due to the metabolic conversions into the inactive forms, which are rapidly excreted, and majorly the removal process is excretion. So metabolism and excretion together is known as the elimination, and hence clearance is in a way defines how the drug is being cleared from the body, removed from the body. There is various types of parameters used to designate clearance, and hence these three things. One is the absorption process kinetics and the distribution, the way in which it is distributed, and the other is the clearance, how it is being cleared from the body by metabolism and excretion or overall elimination. All these will determine what is the plasma drug concentration. We are frequently using only plasma drug concentration term because the concentration which we can follow quantitatively is only with respect to plasma only. We cannot take out a tissue and estimate the concentration of the drug that is available in the tissue. And hence all these understandings, okay, modifications, manipulations are done based upon the plasma concentration only. And hence the plasma concentration or blood level curve is at most important in the area of this pharmacokinetics. This is what is shown on the slide is a typical blood level curve following the oral administration of a drug. And you see on the y-axis it is the plasma concentration, x-axis is with respect to time, as the time progress after administration. Now you see the plasma concentration. Normally, the plasma concentration is divided into various ranges, okay, in relation to the getting the therapeutic or toxic effects. So for each and every drug today, it is well established that there will be a minimum effective concentration. Initially, this MEC is thought to be of only for antimicrobial substances during the development of sulfonamide drugs during the Second World War, when there is a great demand for anti-infective drugs. So during the quick development of this, it was observed initially that a number of compounds, they are not effective in fact. Then they investigated and they found that in the body they are not generating the effect minimum concentration required, that they termed as the minimum effective. Then they postulated a theory that there shall be a minimum concentration required to be effective a drug. Initially, they thought for a number of years it is applicable only to antimicrobial type of drugs, but later on, with the developments in the science, pharmacological thinking, etc., it is proved. Today, it is well established that whatever is the drug, for each and every type of drug, a minimum concentration is needed to be effective, to elicit its type. That is one of the minimum effective concentration. And above that only you find the effect. That means initiation will be there, then it goes on increasing, and then as time progress, okay, the concentration also slowly build up because it's given orally, slowly increases and reaches a maximum. That is known as the Cmax or peak, then again declines because of the elimination. Elimination will always be there right from the beginning, but initial stage you will not find, increasing only. The reason is initial periods after administration, the rate of absorption will be more than the rate of elimination. With the result, concentration will be built up in the body, reaches the peak, and then decreases gradually until the curve goes on, declining curve continues until the drug is totally eliminated from the body. And before it is eliminated, it should produce the desired effect. And hence, the time at which it crosses the MEC after administration, it is taken as the onset of action. For certain drugs, you need immediate effect. Okay. For all drugs which are used to cure acute disorders, like a pain is there, headache is there. You want to get the quick relief. You need not have to wait over a long period of administration to get relief from the headache. So immediately within 5-10 minutes, I need a relief. For that, what is needed is the drug should absorb fast, and hence for all those acute diseases, drugs under the category of analgesics, antibiotics, anti-infectives, anti-inflammatories, all such drugs used for acute conditions, you need a rapid onset and a fast absorbing system is desired, and quickly the concentration is reached. Then if still absorbed, the peak will peak will be increasing. Then you see the y-axis plasma concentration. There is another bar designated as MSC, that is maximum safe concentration or minimum toxic concentration. Okay, so that is the above which you find toxic symptoms and below which it is effective. And hence, based upon the concentration, the three regions are identified. The below the MEC, it is subtherapeutic. Between the MEC and MSC, it is therapeutic. Ideally, you need a concentration in this range only, and it should be achieved as early as possible and should be maintained over a longer period or during the entire treatment period. A concentration within this range, known as the therapeutic range. Then the treatment is said to be very effective, and hence, for several types of drugs, which is normally used for chronic disorders over a long period of time, they need not be immediately absorbed and gives effect immediately within 10 minutes, 5 minutes. But when once to aim to achieve the as early as possible the desired concentration, then the same concentration has to be maintained over a longer period of time in the therapeutic range. For that, today modern delivery systems like sustained controlled novel drug delivery systems are all available. And the concentration is again increasing, you find the toxic, and hence the toxic concentration should not be achieved. So by analyzing the plasma concentration curve, we can decide whether the onset is rapid or not, whether how long it is maintaining the effect, and what is the intensity? Within therapeutic range, the intensity is directly proportional to the concentration, and hence you can evaluate which is effective, which is exhibiting more intense effect, etc. For all such purposes, these curves are very much useful. Now, it is also a curve, plasma curve only, but the case is here different. So the same drug, the drug is not an imodipine, an antihypertensive drug, cardiac drug, given by three different routes: oral, nasal, IV. And the oral means the lowest curve is oral. And virtually, there is no noticeable levels at all. Very meager. They are not at all adequate for producing therapeutic effect. And hence, this drug is oral is not a good route, oral route. The reason being, it is highly insoluble drug at all pHs, and hence the absorption also depends upon the solubility. You need a minimum solubility. Both for good absorption, you need both active solubility as well as the lipid nature, lipid solubility, and a balancing should be there. That is otherwise called the partition coefficient. Partition coefficient, O by W type of partition coefficient is ideal needed more towards lipid solubility should favor, but at the same time, there should be an active solubility also. Otherwise, it is not possible to get it absorbed. So in this case, because of very low solubility actually, it is not absorbed properly, and hence there is no use of giving it by oral route unless you do some changes in the drug or drug formulation and make it more rapidly absorbable by modern ways, means then it may be useful to give it by oral route also. And see, the IV high concentration starts from the top only, because all the drug is placed in the intravenous, so the concentration starting and rapidly declining. Only decline phase is in between. You have a nasal, so nasal is also a very good site of application of drugs, and good absorption occurs, and hence slowly it is absorbed, and the curve is more ideal and suitable for variation in the concentrations are less, and hence for this drug, the nasal drug delivery is more suitable. Like that, the route of administration will also influence therapeutic performance, and you have to select, the pharmacist has to select the right route for its administration. This is to illustrate that concept. And then here there are two curves. Both are oral only. And they are both producing good effect, FOMEC, and not reaching toxic concentrations, therapeutic range, but both the same drug in different formulation, A and B, A and B, but both are suitable. But even then, you find a difference in the blood level picture. In terms of, that means the onset of action will be different, because MEC reached differently in the two formulations. Then the peak is different. Then the decline time also varies. In the case of this formulation, A, very rapid absorption is there, simultaneously rapid elimination is there for the same drug. Whereas in the formulation B, you slowly absorb, reach the concentration, sufficient concentration, and decline is also slow. And hence it will be more a suitable formulation. So formulation influences, formulation differences will influence the therapeutic effect greatly. All the therapeutic characters will be altering. Like these plasma concentration curves are very much useful. And this is another same type of concentration time curve. But here, today, in the earlier case, it is after single dose. But normally, you know that drugs are administered repeatedly. Repeatedly, the drug is administered at the same dose or at different doses. Normally, at the same dose you can try, but several times the initial dose will be high, a loading dose to build up the desired concentration. Then the other doses are a maintenance doses to sustain the concentration over a longer period of time. Here, two cases are considered in this graph. One is then IV injection, 50 mg per hour, okay, of theophylline is the drug given on a continuous infusion. That is one thing. And the other is the oral 300 mg, 300 mg every 6 hours. And when given orally or injections also repeatedly or continuously, what happens is, so slowly the concentration will go on increasing. Here, what concentration is? The plasma concentration goes on increasing until a steady state concentration is reached. What is the steady state is when the input rate is equal to the elimination rate, then the concentration virtually stands at the same level. That is said to be a steady, constant is reached. And when drugs are administered repeatedly, the same drug is administered, it requires 3 to 5 doses to achieve the steady state concentration. And hence, until then, the concentration will be varying. And hence, to avoid that, initially a loading dose will be given to achieve the desired concentration, which is also equal to steady state concentration as early as possible. Then the maintenance dose is given and to sustain the dose within the range. And that is it technique followed. And when the drug is repeatedly administered, you find the peak and valley changes, type of changes in the concentration, like continuously will be getting increasing, decreasing. The dotted line reflect the increase and decrease, increase. That is because it's repeatedly given, and hence you find that ultimately after 3 to 5 half lives it is stabilized. So the stabilized afterwards it is the concentration. The central value of that is shown as the bold line. It goes on increase, stabilizing. And in the pharmacokinetics, when the drugs are repeatedly administered, the word concentration refers to the steady state concentration only. All the interpretations are to be made in terms of the steady state concentration. And the pharmacist should aim at getting the steady state concentration immediately or as early as possible, and then maintain the steady state concentration at the desired level until the purpose of administering the drug is fulfilled. And hence, the steady state concentration concept is very much useful in kinetics. And then linear and non-linear kinetics. There are two types of kinetics, pharmacokinetics, linear and non-linear. In a simple sense, the linear kinetics means the overall elimination is obeying the linear equation. Linear means first order elimination kinetics. Okay, first order elimination kinetics. Okay, and the rate is independent of the concentration. Okay, and linear. Otherwise, the rate is directly proportional to concentration. Okay, the rate is, the more the concentration, the more will be the rate. That is linearity. So a simple test of linearity is that if you administer the drug in different doses, see that in the x-axis the dose is given 200, 400, 600 like that, increasing. Then this steady state concentration is measured and plotted, and hence the steady state concentration is goes on increasing with respect to the dose. As the dose increases, the concentration also increases. That is the character of the linear pharmacokinetics. That means the bold line represents the linear kinetic, whereas the dotted line, so up to some time it obeys linearity. As the dose increases, the concentration also increases, but later on there will be a much increase in the concentration. Okay, much increase in the concentration, though when the dose is increased. And hence that means that here somewhere in the elimination process, some saturation has occurred, and hence the drug is not removed in the same fashion after a certain dose, and hence the concentration is built up. The concentration is increased. That is the reason. And hence this is known as a non-linear. There is no linearity between dose and concentration. So non-linear kinetics are applicable. For majority of the drugs, majority of the drugs, the overall elimination is obeying the linear kinetics only. Means first order kinetics, the bold line is applicable. But in a few cases, in 5 to 10% cases, when in the elimination process, when there is a saturation phenomena dominating, so saturation kinetics are applicable in all the enzyme reactions, and saturation kinetics are applicable in the transport mechanism known as the active transport mechanism. When these mechanisms play more role, more part in the elimination process, where is the scope for this transport? Yes, in the elimination also, there is a mechanism involved, particularly the elimination excretion through kidneys. It also involves the crossing the molecules across the biological barriers in the tubules at two stages, the filtration stage as well as reabsorption. And hence during these process, normally for several drugs, they occur by passive diffusion only. But in some cases, when an active transport mechanism is involved or when a saturation kinetics are involved in this transfer, the overall elimination is also not obeying the linear kinetics, and hence it is said to be non-linear pharmacokinetics. So like the two different type of kinetics are there, but many times it is only the linear kinetics are applicable. It is a simple case to understand further the pharmacokinetics. Okay, this is for this drug when the dose is given in different doses starting from 100 to 50, 500, 1000 mg dose, and concentration mg per ml in with one drug in the first column and with the second drug the second column, etc. Now the question asked is to check which type of kinetics are available. So by simply looking at the data itself, we can say that you see the first column when the dose is doubled. When the dose is doubled, where it is doubled in the third and fourth, fifth, 250, then 500, then 1000. Each time it is doubled. And the concentration, steady state concentration, 37 starting with 250, 37.5, then 75, then 150. Almost it is also in the same order, it is doubled. So that means it is a linear pharmacokinetics is applicable for this drug. Whereas in the other case, you see the corresponding value is 250, it is 62.5, and then it is 190, it is 510. That means it is not obeying the linearity principle, it is abnormally increased, and hence it is somewhere at higher concentration. It is a saturation mechanism or non-linear pharmacokinetics are involved. In that way, we can, by looking at the concentrations itself, a series of concentration, the pharmacist can make immediate inference. And then pharmacokinetic models. And as I said, the basic type of different types of models are used, but pharmacokinetics is a compartment model. That means the body is divided into various segments or compartments. There is no physiological relevance to this division. Okay, but it is this division is made based upon only the kinetic behavior. So kinetically distinguishable pool is otherwise known as a compartment. So a group of organs or tissues into which the drug is entering in the same way and leaving in the same fashion, all those can be grouped as one in pharmacokinetics. That is called a compartment. So all those which organs and tissues which are behaving in a different way in receiving the drug and leaving the drug, it is considered as another compartment. Like that, based upon only the kinetic behavior, how each organ is receiving the drug and leaving the drug. Based upon these two things, and rate of entry, rate of leaving. Basing upon these things, the body can be divided into various segments. And most widely, one compartment, two compartment, and multi-compartment models are used. And there is no need to go beyond that. And unless it is a highly specialized study of examining the behavior, where the drug is going, whether it can enter into a specific region in the body to serve any purpose, etc. And those special cases, other than that, generally the 1, 2, and in general treatment, multi-compartment model treatment is adequate. The drug may be given by any route, but these 2 compartments are adequate. And these models can be used to predict the time course of the drug. And hence time course, what is happening to the amount and concentration of drug in the plasma, in the tissue, or otherwise the first compartment, second compartment, and in general the compartment that are issued. For that purpose, the mathematical model is generated. So it is based upon that there are certain parameters are arrived at. The raw material for the pharmacokinetic study is the plasma concentration versus time data. Following the administration of drug at different times, the plasma concentration, that means unchanged drug concentration in the plasma, is estimated, and then based upon that raw data, the graphs which we have seen earlier, different graphs can be drawn and various parameters can be calculated, worked out. And these are some of the important parameters normally worked out. One is the elimination rate constant, biological half-life, and these 2 are related. The biological half-life, you know very well, the time required for 50% elimination of the drug from the body. It is biological half-life, is a constant for a particular drug. And clearance is an elimination parameter similar to half-life, but different from that. So here clearance, total body clearance is there, renal clearance is there. They both are on the same concept. So clearance, it is the volume of the body fluids from which the drug is removed per unit time by all organs is known as the total body clearance. In the same way, the volume of the body fluids from which the drug is removed by kidney per unit time is known as the renal clearance, designated as CLT and CLR, which are very much useful to explain the elimination character of the drug. And so these are the first 4, they are referred to the elimination properties, characters. Which are likely to affect the performance of the drug, therapeutic performance, etc. And apparent volume, VD, volume of distribution, is a volume of the body fluids in which the drug is distributed. And it's also a useful parameter, but it's not reflecting the true volume. It's always an estimated volume, hence apparent volume of distribution, but used for in the kinetic purpose. So to equate the concentrations to convert concentrations into amounts, because the doses are given in terms of amount, quantity mg, but concentration is measured in terms of concentration per volume ml. So to convert one into another, you need a volume factor, and then that volume factor is introduced as VD. So Cmax, Tmax, AUC. These are from the plasma curves, peak concentration, time at which peak concentration occurred, area under the curve from 0 to infinity, which is a measure of the extent of absorption, availability of the drug to the system, systemic circulation, and all these are something related to absorption. So absorption rate constant, the rate at which the drug is entering into the body, AA, and bioavailability, it is the effect of both the rate and extent. So the rate and extent of availability of the drug to the system is otherwise known as bioavailability. Again of 2 types, the relative bioavailability and absolute bioavailability. Absolute bioavailability is bioavailability of a drug administered by other routes with the IV, by extravascular routes with IV. IV gives the maximum availability, considered 100% always, because drug is directly placed in the in the vein, and hence it is 100. And compared to 100, the availability of the drug given by other routes is estimated as this absolute bioavailability. Relative means when the dosage forms, like in the earlier graph, formulation A, formulation B, and to estimate the relative bioavailability, one which is having the larger area is taken as the 100%. The other formulation, its bioavailability is expressed as percentage of the maximum. Or if you have a standard product, okay, these two test products are compared with the standard product or innovator product, especially in the design of the dosage forms. For such comparisons are needed for ANDA application. So you need to compare relative bioavailability and compare with the innovator product and bioequivalence. So especially again a concept very much useful in practice when you want to change. The drug is the same, but product changes. Product changes frequently. Here, product changes normally goes at the whims and fancy of the physician. If he likes some drug, the company, he will prescribe that. But no one sees about the quality of the product. By name only, we go largely in India. But whereas when the pharmacy practice is well developed in all other areas, the pharmacist is competent to decide which brand, which product of the drug has to be used for his patients. Okay, and that concept, that doctor will not interfere. He will suggest it to the pharmacist, only clinical pharmacist or registered pharmacist. He is capable of judging the formulation essentially based upon the concept of equivalence. And after establishing the equivalency between products, then the pharmacist can easily make any change in the product to be administered. Like all the pharmacokinetic parameters are very much useful in the practice as well as in the design, etc. And so they can easily be worked out either manually or using any software. Many softwares are also available today. And applications of pharmacokinetics. First, we'll see the overall, what are the general applications of these pharmacokinetics means. So this data, this is applicable at several stages, particularly in the design and development. It is useful. How it is useful, why it is useful is, now the drug should have sufficient aqueous solubility, at the same time lipid solubility. These characters can be manipulated after making the NCA, new chemical entity, many times that do not possess the adequate kinetic profile, pharmacokinetic profile, unless you make changes for altering the lipid nature. Okay, so partition coefficient has to be worked out exactly to match one. One means equally polar, equally lipid. And hence the best, very rapidly absorb. One such drug is alcohol. Alcohol is, you know, that is equal miscible with water and oil. So that is the reason why alcohol after administration orally, immediately you will find the concentration will reach in the blood. Okay, immediate effect. And so these type of adjustments in the lipid solubility, aqueous solubility will be made by using the concept of the concepts developed in biopharmaceutics only. And then in the same manner, in the design of dosage forms, all types, particularly in the modern controlled delivery systems, which control the rate of entry of the drug into the systemic circulation, and in a controlled fashion, the system will allow the drug to pass on to the blood and over a longer period of time. So in all those formulations, it is very essential that the elimination kinetics are to be utilized and do in dosage calculation and achieving the desired rate, etc. So in the design of these modern drug delivery systems, it is very much useful. Then to just know the dose, to estimate the dose. Okay, dose has to be first initially worked out in experimental animals in the preclinical, and in the phase 0 studies, the dose has to be standardized in the humans. And these things are all a part in the pharmacokinetics only, that how to calculate this dose, and particularly this loading dose and maintenance dose. Some of these issues we will see, and dosage regimens, particularly in diseases when the elimination process is much affected. Many times, many diseases, the problem comes with the kidney, renal function is impaired, or liver function is impaired, damaged, or with respect to age, or with respect geriatric patients or in special cases, all adjustments are to be made. Why you need these adjustments in the dose? This is in the due to the altered physiology of the subjects, the patients, with respect to various same drug will be toxic because it's not removed from the body unless you take care. So only care is that you reduce the intake, cut down the dose. So how much it should be cut down? Those manipulations need and found to be understood by looking at simple parameter known as half-life. And accordingly, with all the drug interaction, drug-food interaction, and various other things that interfere with the drug at different ADME, all these things it is possible by the use of pharmacokinetics only. And clinically, the two important areas are one is the design of optimum dosage regimens in different in patients. That is one. And the second one is clinical management of individual patients. That is therapeutic monitoring, therapeutic drug monitoring. In the cases where therapeutic index is very narrow, the range of therapeutic concentrations are very narrow. So unless you take special care individually, measuring the concentration and rapidly making the changes, many times the drugs are administered by intravenous continuous infusion only. Drug admixtures are prepared and added, and good control is possible instead of using the other types of dosage forms. And the pharmacist should well equipped, and this is all the patients in the hospital, inpatients when it is needed for which type of drug. People, so some drugs, but they are all life-saving drugs, and hence, unless the pharmacist well versed with the therapeutic drug monitoring. So in India, it is not yet fully started anywhere, but in all other advanced countries, hospitals, this concept is well practiced, and we are only theoretically we are sound, but having all the adequate facilities. And also in the hospital also today you need a good analytical instrument to assay the drug. The drugs are very potent. The plasma concentration has to be assessed accurately, quickly, and hence you need the sophisticated analytical equipments. And without which, okay, it's not possible to implement this what is called therapeutic drug monitoring. So we expect a day will come. Such things are also being established well in India. So further, the pharmacist has to prepare themselves and to face the challenges by undergoing adequate training, if not here, elsewhere. And then this is a one compartment open model. Just a few cases only, and that too superficially we will consider. And this is the pictorial representation is like this. There is, it is the entire body is consisting as only one compartment. That what it means in essence is the drug is distributing to all organs in the same way as it is entering in the plasma. The moment it is entering into the plasma, next moment it is also available in the tissue. That means the distribution is very, yes. Don't say there is no distribution. Distribution process will be there, but it's so instantaneous. Its effect cannot be worked out mathematically. Okay, and the reason is that immediately, instantaneously, drug crosses the barriers and entering the tissue, and the same concentration as it is in plasma is achieved in the tissue. If not mathematically the same figure, but the ratio will remain the same constant. So ratio means the concentration of the drug in plasma divided by concentration of drug in the tissue, Cp by Ct, is equal to a constant K, which is ideally 1 or it is 1.1, but always it maintains in the same manner. Okay, and hence instantaneous distribution will be there. So in which case the concentration is indicated by Ct, and the mathematical model is Ct is equal to C0, concentration achieved immediately after injection, and in the case of IV, and then exponent. This exponent is a mathematical constant referring to first order kinetics, obeying the linear kinetics, and to the power minus KELT. KEL is the elimination rate constant. And hence, in this case, there is only one rate process involved. It is all elimination phase only. Means because a drug is administered by IV, entire drug is placed, dose is placed in the blood plasma, and otherwise only decline only. Distribution is instantaneous. So right from the beginning, the curve will be declining. Right from the beginning, the curve will be declining. And hence there is only one. So from this, okay, it is converted into logarithmic equation, and then equations are there for estimating the half-life and other quantities. Okay, and then this is in the same type of curve, the same system, following IV dose only, when a logarithmic curve, because first order means the variable is not C exactly, log C. Log C versus time, if you plot a graph, it will be a linear graph indicating that the elimination is obeying the, because there is only one process elimination. Whatever curve you get, it is all representing the elimination process. And then the slope is calculated like that. Slope is equal to minus KE rate constant divided by 2.303, again a constant. And then using the slope, you can find out the biological half-life. This, okay, slope K is equal to slope into 2.303, and then when once KEL is known, the T half is again 0.693 by KEL, simply you can calculate. So these manipulations, okay, you need not do always the graph. Okay, even if you know two concentrations, still you should be able to calculate half-life, because drawing a graph and finding slope and all these manipulate requires time. That you can do it in a laboratory, but in a hospital it is not. By sitting or standing by the side of the patient, if you have the two samples, quickly analyze and you get two concentrations at 2 times. Using that, you should be able to find out. You should be able to find out the slope and half-life quickly within a few minutes. Then that information has to be used in making the adjustments, and hence this is the normal way. And easily the other things are when one relations exist, like VD is equal to X0 by C0. X0 is the dose administered amount. The entire amount will be there in the body at 0 time only. If you wait for some time, something is eliminated. What quantity is eliminated is very difficult to estimate, and hence X0 by at the same time 0 time you should know the concentration. That is not possible, and hence the method is known as extrapolation method. So this type of curve is the slope is identified, straight line is identified, then extrapolated back to intercept y-axis at the C0. The intercept is log C0, take anti-log, it is C0, use it here and calculate. This is all is easily, you should be able to do as quickly as possible these simple manipulations. And then this is with the same one compartment, several times it is useful. Then here you have the oral dose, extravascular dose. Extravascular dose is given. So this is the case where there are two exponential terms. In the model equation is C is equal to a complex figure multiplied by two exponential terms. One is related to the elimination, another is related to absorption. Because in the pictorial representation, you see that X is outside the compartment body, then it's slowly entering into the body, and the rate is associated, Ka, absorption rate. Then it is distributed, and then it is ultimately eliminated from the same compartment, one, and elimination rate is KL. And hence two processes are involved here when the drug is given by extravascular route other than IV. And hence the equation is two exponential terms are there. And using in the same manner here, you have the expressions developed for Cmax, what is Cmax, and Tmax, etc. And these are all just, it is enough that you know about Cmax. So Cmax, the expression given for Cmax is second one from the bottom. Cmax is equal to F into X square, X0. X0 is the dose given, administered orally. But all the drug administered orally may or may not be reaching the central compartment. Only a fraction will reach. That fraction is otherwise F. It is otherwise known as absorbable fraction, bioavailable fraction. In the earlier bioavailability concept, we have seen that it is otherwise known as the absolute bioavailability, which is estimated against IV dose, comparing against IV dose. That F value, okay, into the X0, that gives the amount really entering into the plasma when given orally, divided by VD, because that is amount, divided by VD means concentration, and multiplied by exponential term, it will be decreasing. Initially, it will be there and it goes on decreasing with respect to time according to the KEL. And hence, mathematically, the Cmax depends upon the dose administered. The more the dose, more is the Cmax. More is the F, the more is the Cmax. And VD, VD. And here, if the drug is distributed into large volume into each and every part of the body it is going, then normally the Cmax will be very less. VD in the denominator, and hence the figure is less. Okay. And VD, large value of VD does not always indicate large distribution of the drug. Sometimes if the drug is localized, selectively stays in certain areas, a lipid soluble drug may conveniently settle in a fatty depot. Okay, and hence it is settled there only, but not available to the plasma. In the plasma, little drug is there, and hence concentration is little. When concentration is VD is low, okay, VD is concentration low means VD is large. When VD is large, the Cmax appears to be very less, but it is an erroneous. And hence, careful interpretation has to be made with regard to the VD. And the purpose of VD is only as like this, a proportionality constant to equate amount with the, here itself, amount with the concentration, Cmax and X0. To equate it only, you need the V. And hence, in all the kinetics, it will appear, though it has no much significance. This is again oral only. The same data when drawn on a semi-logarithmic log concentration versus time. And hence here it is a biphasic curve. This is the first elimination phase, and to resolve what is the absorption phase, there is a method known as the method of residuals or feathering. And hence there are techniques to understand that it is with the help of which the data is a sum of the absorption, and in the absorption phase, it is due to absorption as well as distribution, as well as elimination. And hence to resolve these things, now a technique called feathering is applied, and that feather line or residual line, it represents the absorption process, whereas the original dot thick line, it represents the elimination process. There is no distribution. Why distribution? No distribution means it is so instantaneous, because we are considering only one compartment model. And when the blood data is available, if we draw a plot like this, and if we extrapolate the linear curve backwards to intercept the y-axis at C0, this C0 cannot be the same as C0 in IV dosing, which is entirely different. In IV dosing, C0 is used to calculate VD, but here it cannot be used because this is a complex factor involving the influence of F, X0, VD, and the relative magnitude of Km and KEL. That's what the log A is equal to, so much big factor is there. So many factors are available, are entered. So in the mathematical equation, so you have, this is oral means two phases will be there, two exponential terms, one related to absorption, two the elimination. And again, distribution is instantaneous. And we have seen already F fraction absorbed. Similar curves you have to generate following the IV dosing, and hence AUC, area under the curve, following the oral, divided by AUC, following IV, keeping the dose same. and hence the F can be calculated. But the problem is the oral doses are large, and when compared to IV dose, you cannot do the same amount. So dose will be different, but each area should divide by the corresponding dose to apply a correction. And then, so AUC oral by that oral dose. Similarly, AUC IV by IV dose. Take again a ratio, it is known as the F. So like that, absolute bioavailability can be assessed following the oral route if the dose is different. So this is a simple case, but a very interesting case. The name of the drug is gentamicin, which is the use of this as antimicrobial, okay, antimicrobial, and it's very much used to treat deep infections, bone infection and blood-brain barrier infection, spinal cord infections. So deep infections, but this drug is very powerful. And the drug is used at a very low dose, 2.5 mg per kg only, and it has a severe toxicity. It has severe toxicity and neurotoxic. It is neurotoxic, and it's also affect the kidney patient, the liver function. But when it is recommended for liver, for the kidney patients also, means to treat all the infections after a few doses, they get reduce its efficiency, renal inefficiency will work out, and hence the doses are to be carefully titrated. So to illustrate that, it's a very interesting drug molecule. Similar drugs may be there in the practice also, and this is one of the most widely used antiseptic drug, antibiotic drug. Dose is less. Its half-life is only 2 to 3 hours half-life. That you remember. Now, they in one study in a hospital, so time after administration, this is normally given as infusion only, okay, slow infusion, and concentrations are by concentrations estimated after the time, 1 hour, 24 hours, 48 hours. So in experimental investigation, you can collect samples hourly like that to use several purposes. But in practice, it's not possible. It's enough only to take a sample. Okay, occasionally 24 hours and 48 hours, because continuously it has to be treated, and then there is no time to draw the graph and find out all the logs and logarithmics, etc. No, nothing. So quickly you should work out half-life, because half-life will reflect, half-life of this drug will reflect 39 per hour, inverse hours. Elimination rate constant, substitute simply that T half is equal to 0.693 by KE, and which is worked out to be 50 hours. 50 hours. That means by the time this is 50 hours. Now, in the normal patient, it is only 2 to 3 hours only half-life. 2 to 3 hours. So it is rapidly eliminating. It's rapidly eliminating drug, and rapidly eliminated by urine only. And hence, such a drug which is supposed to be rapidly eliminated, the half-life is very much elongated, 50 hours, based upon the data. Okay, and hence, what is the reason? The reason is that the kidney is affected, kidney function is affected, and the kidney function is not properly doing, means that the major route, major process of elimination of this drug is only through kidney elimination, excretion. That is impaired, and hence the drug is accumulated in the body. When drug accumulated in the body, the half-life increases. The half-life increases normally. That is the reason why. Otherwise, the half-life is only 2 to 3 hours it is given. If not 3, you may expect 3 to 4, 5 hours, but not so much, 50 hours. Means that the kidney renal function is greatly impaired. And if the treatment is continued like that without monitoring, it has drastic influence, because it is a neurotoxic drug. Neurons, nervous system will be affected, and hence all the functions will be lost. And hence in such cases, now Like this, in the diseases, so first is understanding the effects of the drug, the possible toxic effects of the drug and behavior of the drug in pharmacotherapeutics, okay, how it behaves, etc. Okay, and then the pharmacist should be able to judge the function of the kidney, function of the liver periodically, and hence other parameters are to be worked out. The another important parameter apart from half-life is clearance. So these are the two things in a patient. Clearance has to be followed more carefully. Okay. Or half-life has to be followed more. Half-life is rather easy than clearance. So to estimate clearance, you should know. So once the half-life is known, the clearance can be estimated using the mathematical relationship between the two. That way you can do it. And hence, so the data also suggests in the previous slide, so 48 hours, the decline is from 7.7 to 4 only, not even a half. The decline is not even a half. And hence, so by looking at the data itself, you can suspect, but when you work out, the real thing will be known 50 hours. What is how we defined half-life? Time required for 50% reduction in the concentration or amount. So 7.7 should become concentrate half of that means 3.8 or something, but it is 4 only. If it becomes 3.8, it will be 48 hours, but it is 4 only, still above, and hence more time, 50 hours is the biological half-life. So now this parameter like this, if we estimate it simply by measuring only 2 or 3 concentrations, is it necessary to do in each and every patient? Need not be. Only in the cases where the drug is critical and its side effects are critical, influencing the vital organs. In such cases, necessarily have to monitor whether the drug is eliminated properly or not. If it is eliminated, there is no problem. Okay. But if it is accumulated, then only toxicity will increase. And hence, it suggests that immediately you have to go for change in the dose. Now, why half-life is important? A simple parameter, but it's very important because it gives the duration of action after a single dose. Normally, the drug is effective over a period of 2 to 3 half-life times, not more than that. You have to go for a second dose, and the time required to reach the steady state. So we have seen in the beginning, steady state concentration should be reached early, and hence the half, it depends on the half-life. If the half-life is short, it reaches quickly. If the half-life is long, it takes still long time to reach the steady state. And then the dosing frequency. If the half-life is important, if the half-life is very short, dosing frequency will increase because rapidly drug is eliminated. And hence half-life gives much useful information like this. And then estimation of half-life was seen already, and hence the simple equations, relationships you find, you should keep it ready. Whenever you need, you can work. So CL means clearance. Clearance is the volume of the fluids from which the drug is removed per unit time. That is, traditional method involves estimation of the drug in blood as well as urine and finding out the rates of rate of elimination, rate of excretion, etc. In addition to concentration, and otherwise, KEL can easily be observed as seen in the earlier case. Using that KEL information, knowing the VD, you can simply get the CL. Okay, and then CL, in this case, the other case, it is equal to the relation. These relations, the pharmacist should be familiar. After working out a few cases, you will be knowing that, and easily you can manipulate, convert one thing into another easily. After 4 half-lives, 94% is eliminated, 94 hours. So that's why 2 to 3 half-life times is adequate. It produces effect. After that, you will not get any effect, and hence you should give a second dose like that. And more exactly, what doses are given, you have to find out. This is again the drugs with short half-lives reach steady state rapidly. We have seen Vice versa. Drugs with longer half-lives take a long time to reach steady state concentration. Until then, the concentration will be fluctuating. And why? What is the disadvantage of fluctuating concentration? The concentration they are fluctuating means they tend to precipitate the unwanted reactions. Side effects will be more precipitated if you allow wider fluctuations in the concentration over a longer time. That is the reason why the modern formulations like sustained controlled release formulations are needed for several drugs, not necessarily short half-life drugs, but for all drugs it is very essential to see that fluctuations will maintain, will be removed, and ideally same concentration, and hence side effects will not appear much. And so this is one of the important principles which you have to remember. And then long half-life, the consequences you already seen some, and easy to maintain the plasma levels within therapeutic. It is rather easy with drugs having long half-life. With the half-life we start too rapidly, it is declining, and hence frequent dosing is needed. Missed doses are no big deal. Means that though some doses missed, nothing will happen because long acting. All these are long-acting long-acting drugs. So some of the drugs that are described as a long-acting means the half-life, based upon half-life, only half-life will be more. Half-life more means it is eliminated rather slowly. Inherently, the drug molecule is designed in such a way that it resists the biotransformation and excretion. With the result, it stays in the body for a long time. Long-acting drugs. And dosage form wise also the duration of action can be increased by dosage manipulation. But inherently the API itself, some APIs they are called long acting forms. By altering the chemical configuration, it is possible. So a disadvantage also there, if toxicity occurs, it is a long wait. The toxicity will not appear immediately, but after a long time only you can see the toxic effects if at all it is there. And that is only disadvantage. And initial therapeutic effect developed slowly without a loading dose. That is the reason why. So for drugs having longer half-life, the initiation takes long time. And in such a case, you need anyway, modern way is whether it is long-acting or short-acting, we will add a loading dose. A loading dose, the purpose is to initiate the concentration as early as possible. In today's pharmaceutical manipulation, the loading doses are separately treated and manipulated to see that enhance the absorption and get to the concentration immediately. And then once it is achieved, and then it is sustained over a longer period of time by other manipulations. Like this, by pharmaceutical manipulations, this can be tackled. Short means the onset of therapeutic effect tends to be very rapidly, onset of action will be there. And so if toxicity occurs, it is not a long wait. Quickly is also being removed. Where drug is rapidly eliminated, and hence toxic effect noticed will also quickly be avoided because the drug is eliminated itself rapidly. And this is about the volume of distribution. We have already considered this is just a parameter. Volume of distribution means amount of the drug in the body by C is the main use of this volume of distribution parameter is in calculating the loading dose. So loading dose, how the loading dose are to be worked out is you have to find out the CSS, steady state concentration. Steady state concentration multiplied by VD is otherwise the loading dose. In general, the dose, how to calculate a dose of a drug in the developmental stage. Effective concentration is known, and effective concentration, how to know effective concentration. So minimum effective concentration, toxic concentrations are to be worked out by trial and error methods or by dose, goes on increasing the doses in animal species until and record the response. So you will find the dose, effective dose and the toxic dose, and the central point of these two things are known as effective dose is taken initial stages. Then using that effective dose, or the concentration, calculated CSS divided by VD is otherwise known as the. So in experimental, in the preclinical studies, all these parameters are to be assessed in animals, And in the animals, no problem. If at all the animal has to be sacrificed, you can do it and estimate. But it's not possible in humans, and hence in the initial animal experimentation is also very much needed in the drug development process to assess all the pharmacokinetic parameters and which form the basis for zero phase initial trials in humans, small number, not more than 10 to 15 subjects, and to standardize some of these parameters, useful parameters in subjects. And if it is all new drug, it is a old drug, certainly a lot of literature is available on an anesthetic agent at a rate of 2 mg per hour by IV infusion. The elimination rate constant is given here, and the volume of distribution is also given as 10 liters. And what loading dose should be recommended if the drug level to reach 2 microgram per ml immediately. Okay, immediately. Such problems will come normally. The doctor will may ask the pharmacist to tell him quickly. And so here it is, all the given data is written on the top. Infusion rate 2 mg per hour, elimination rate 0.1 inverse hours, volume 10 liters, and required constant 2 microgram per ml. Then loading dose we have seen VD. VD should be known. To check whether VD is there or not. Yes, 10 liters. And there is okay. And CSS is 2 microgram per ml, and it is equivalent to dose is equivalent to 20 mg. So simple manipulation only, but rightly you should understand the concept. So when loading dose is asked, immediately it should come to your mind that it should be based upon VD and CSS only, and hence easily you can check for those. And clearance. So the concept of clearance is very much useful for maintenance dose. For maintenance dose, this concept is very much useful. So clearance into CSH is otherwise known as the maintenance. So what is maintenance dose is whatever is removed, you should be able to replace to maintain the concentration at the same level. For this purpose, it is based upon the clearance value multiplied by CS. If the CS is multiplied by VD, it gives only loading dose, which is different. Maintenance dose is different, and hence, apart from the CSS, steady state or desired concentration, the other parameters which you need is one is VD, another is CL, clearance. If you know these things, you can easily calculate these doses. And it is quickly we will rush to some of the cases, but it takes more time to clearly see all the information. So only in a nutshell, I will tell the equation that best fit the plasma level time curve, okay, of a drug that is given as C is equal to mathematical equation is given, calculate. That means we have seen the model means the mathematical equation that describes the model, that describes the C. C means the variable. The concentration is varying like that. And hence you are asked to find out what is the purpose, what is the most interesting here is when should the next dose be given if the drug becomes ineffective when the plasma falls below 50 microgram per ml. Like some intrinsic questions will be asked, and now again it will come. So first case is apparent volume of distribution is given the CD. Okay, this is the equation. C is equal to 143 exponent minus 0.87 T. So here it is exponent. Remember the earlier equation. 143 refers to C0 and minus, because it is an elimination process, minus, and quantity is 0.87, that is the KL, and hence you can use that information and find out dose is given and VD is worked out simply. Okay, similarly, elimination of life also can be worked out. So sometimes in the literature, instead of giving the entire kinetic data, generally the mathematical equation, if it is given, that is the purpose. Mathematical equation is a concise way of telling the time course of changes are happening. And hence, if the equation is known, you should be, as a pharmacist, we should be able to find out essential parameters. So if that above equation is given and you are asked to find out what is the half-life. So the half-life is in the same manner. Ke is that 0.870 and hence 0.693 by this is the half-life 0.8 hours. So that is the purpose of using the, don't expect that every time you will get in the literature the exact data. Data is not needed. Only thing is the entire data is condensed into this mathematical form only, and you should be able to understand how to analyze, work out the things. Plasma concentration after 6 hours in the same manner. So the amount of drug that will be left in the body after 6 hours. So all these manipulations only to understand the equation clearly it is possible. And then more important things only we will discuss because so many things are there. This is another equation, same thing, 87, and another question like this, it continues. And this is the kinetics of multiple dosing. Kinetics of multiple dosing. Drugs are repeatedly administered. Repeatedly administered means you find a peak and valley pattern, increasing, decrease. That should be avoided. And hence infusion is one way and then sustainedly the another way like that. But still the multiple dosing is followed. Drugs are repeatedly administered for ease. Practice even today, drugs are repeatedly administered. Asked to be taken every 6 hours. Okay, every 8 hours. 2 times a day, 3 times a day, like the instructions you see in the prescription. Today it is much widely used. So this is the very curve, plasma concentration curve like this. And then, so there when you administer the drug repeatedly, the same drug, CSS, steady state concentration, average, the formula is Fx0 by CLT and tau value. CLT in the denominator of, that is not T tau. Tau means the time interval. And this is in the same manner. If you convert the same equation, this is average. Average. This is for amount x. X stands always for amount. But instead of amount, we should use the concentration. The first term is more appropriate. But both things can be converted. Only difference is that you should divide it with VD. That's the only difference. And the other conversions are to convert into various other forms in terms of VD, fraction absorbed, etc. Okay, it serves. But essentially, the average concentration will depend upon the dose given and F is fraction absorbed in the case of oral and clearance and tau value. Tau value means the time difference between the two repeated administration, time interval. If the time interval is large, the concentration will be less. If the clearance is large, rapidly cleared, eliminated, CS is low. Okay, like that, you have to be using that equation, you can make inferences. If the tau, so tau should be appropriately evaluated. And hence, the more important thing here is how to find out, how to fix. The dose adjustment is based upon the clearance concept or T half-life, biological half-life. It is one dose adjustment based upon the total body clearance. Okay, the equation is this is the average steady state concentration to be kept normal or in the normal patient, and F is the, this is assumed to be constant. This is depends on the route and the type of formulation, etc. Into this is this side is again 1 by CLT clearance decreased due to disease into again x0 by tau value. Tau value is the time interval. Okay, and using this, that means if CL clearance dash and x0 dash are representing the values for the renal failure case. In the case of renal failure, what happens? The clearance, what happens? The clearance will get decreased. So CLT decreases in the case of renal. And in such a case, the equations can be rearranged to find out the modified dose or modified time interval. That we will see here. Okay. Okay. Now we'll see this with an example. Instead of reading the equations as such, you can easily understood if you work out a simple case. So this is a drug again administered to a 70 kg patient at a rate of 2 mg per kg every 12 hours by multiple intramuscular injection. The drug has a half-life of 2.2 hours and VD of 0.2 liters per kg. This is the data. Determine the CS maximum concentration, CS min and CS average. More important is the CS average. You see where is CS. Other things are like this. Half-life is given and KEL is given, which can be calculated from half-life. It is available and VD is given. It is available. Okay. And VD of the patient is 0.2 into 70. 70 is the total weight and VD is given as 0.2 liters per kg, and hence this is the total VD, and X0 is given, the tau value is 12 hours. These are all the given data, and hence you can substitute this data in the equation of CSS max, maximum steady state concentration, and which was worked out to be 10.23 microgram per ml. Milliliter, 10.23 microgram per milliliter. That is the just the values you could organize in the proper way and get the appropriate expression and substitute and work out these things. Then next comes CS min. In the same manner, CS min is 2.3 microgram per ml. So maximum is 10.23 micrograms per ml and minimum is 0.23 microgram per ml because variation. And then average, CS average is worked out using the earlier equation in the same way, and then it is found to be, that is CS average is worked out to be 2.64 micrograms per ml is the average steady state concentration. This is using the data, these are the values obtained. Now comes the case of a patient. So if the T half increases to 5 hours in renal insufficiency, what should be the new dose or the new dosing interval? So these two things are to be worked out. And hence here the dose, the half-life is elongated. Why? Means the kidney is not working properly, accumulation takes place, and hence the drug stays in the body for a long time. So the half-life has got increased. And hence the new dose, okay, you have to calculate. You should not give the entire dose. So how dosage is calculated? And the dose unchanged, dose without, okay, it is done. So new dose T unchanged. You have to keep the T unchanged. It means as it is. Right? It is 88 mg per kg, and new dosing interval, don't change unchanged dose, and it is 27.23 hours. And half-life 2.2. These are the values which are given. And now come to the real calculation of substituting what values. So here the cases are, one is the unchanged dose, okay, and T half in the unchanged case, T half is 2.2 hours, and in the changed half life in the renal insufficiency, it is increased to 5 hours. Okay, 5 hours. So in one case it is 2.2 hours, got increased to 5 hours. And hence tau value in the when the half life is 2.2, tau value is 12 hours. When the half life is okay, increase it to 5 hours, you have to calculate what is the tau value. That is the question. Tau value you have to work out. And hence here, if you substitute those data in here, the tau value is found to be, 27.23 hours. So that means the time has to be elongated. Okay, time. Originally, what is the tau value? What is the tau value originally? Tau value 12 hours. Now the tau value, the revised tau value should be, okay, the tau value should be 27.23 hours. So much elongation is needed. because the half-life is much increased, means the drug is staying in the body for a long time, and hence instead of giving the drug at 12 hours every dose, you elongate further. You give it at after 27.23 hours. That is the way in which one type of adjustment is that this way you can do it. And other type of adjustment you can also make it as, so that means x0 can also be changed. So here it is, half-life remains, half-life is, the original half-life is 2.2, revised half-life is 5 hours, and tau value, both the tau value we want like to keep it as 12 hours only, and hence in this direction the x0 you need to administer, the revised x0 is 8.8 mg per kg. What is the original x0? Original x0 is okay, original dose, and hence the x0 altered is 0.88 mg per kg. You have to that is earlier also given the same thing. Okay, and like this in the adjustment there are two ways. One is first keep the dose unchanged. X0 and X0 dash both should be unchanged. Keep it the same way. And the other way is keep the time interval this change and estimate the doses. Change the dose. In the first case, keep the dose same and estimate the time interval. The time interval is increased. Increase the time interval, you have to revise. In the second case, when the time interval is retained same, the only alternative is you have to reduce the amount to be given, and hence by using these mathematical equations, the relationships which you have to remember, they essentially remember the equation x0 dash by x0 dash by tau dash is equal to t half x0 by t half dash. Dash values means the revised values in the renal failure values, case value, tau value. Okay, and use this equation. Remember this equation, you can easily work out. Okay, this is if the drug is to be injected once every 24 hours in renal insufficiency, what should be the new dose? So it is directly asked. The tau is instead of 12 hours, now it is injected 24 hours. 24 hours. And then what should be the? It is decided to increase the tau value. Drug administration has to be increased 20 to 24 hours. And what should be the new dose? Okay, in such a case. Again, the same equation can be used and substitute the values, and okay, the revised dose is 1.76 mg per kg. So here, such dose mg per kg is needed to sustain the effect, revised dose, and hence these equations will work well in the cases provided you know the values of either T half should be estimated or clearance should be estimated. These are the two parameters which are very widely used in dosage adjustment in renal and other disease conditions. So here it is again a child. This is several times for child. The same drug has to be administered to a child weighing 12 kg here with a normal renal function. What should be the renal function is the same, but because it is child, normally you need a less dose because the metabolizing systems are not fully developed. The body cannot tolerate the larger doses given, and hence a child dose, the best formula that is used in therapeutics is always the weight of the child in kg by 70 to the power 0.7 into adult dose, and is a simple equation, and there may be other equations also well recognized, but many physicians and therapeutics recognize this as more useful equation, and hence accordingly you can make use of this simple manipulation. The dose comes to 3.39 mg per kg. Okay. And weight, adult dose in this case is adult dose per kg is okay. By 12 okay, per kg. And hence, this is another case where the drug is to be administered to an elderly patient of 85 years and 55 kg body weight. Determine the dose. Here it is the age wise only, but on the other side, an elderly patient. Again, the dose has to be modified. And here it is the patient's dose. Here it is called weight in kg to the power 0.7 into 140 minus age in years whole divided by 1660 into adult dose. And in this case again, the same, all the information is available, weight in kg is available. Okay, patient weight is 55 kg and years are available 85 years. And then adult dose is given as 140 and then you reduce it and it is in terms of kg based upon the body weight. So total dose is 76.67 mg or in terms of kg dose 1.39 mg per kg. Like the simple equations are available in therapeutics, in posology also they are available, and more appropriate equations you should select and quickly manipulate to reduce the dose. And it is here again, in this case, again a case with a liver failure. Again, liver failure also needs to be, liver failure means again the elimination, overall elimination will be much reduced. And hence the normal subject's clearance is given as 45 liters per hour and volume of distribution is 175 liters. Liver failure, in that case, the clearance is 15 liters. So it got reduced. Originally it is, the clearance is, original clearance is 45 liters, normal subjects, and it is reduced to 55 liters. And then these normal values, sometimes they are taken, normally they are taken from the literature only, values representing a large population, and hence these values the patient values will be available in with the pharmacist of determination, and then the what adjustment has to be made in this case, we will see. So given information is clearance, VD 300, CS is 10 mg per liter has to be maintained. So first is loading dose. Loading dose should be based upon the VD, and hence loading dose is equal to the VD. VD is 300 liters into the concentration to be maintained is 10 mg per liter, and hence 3000 mg, 310, 3000 mg is the loading dose. It is given as intravenous bolus injection. It produces the desired load the concentration, CSS. Then comes the loading dose dimension is mg, because only once it is given, so in terms of mg only it is given. Then maintenance dose, next is maintenance. So for here the parameter needed is clearance, it is not weight clearance. And the clearance is also given, but here clearance is the revised page in the patient, the clearance is 15 liters per hour. And in the normal patient, the clearance is 45, okay, clearance. What do you understand by this difference? The 45 in the normal patient and this page normal subject, and in this patient is 15 liters. So there, in which case elimination is rapid? Elimination is rapid in the first case normal, because per unit time, so much volume, from so much volume drug is eliminated, 45. And in this case, it only from 15 liters the drug is eliminated per unit time 1 hour. And hence the elimination is very rapid in the normal subject, but in this case elimination is got much reduced, and hence necessarily the doses are to be modified. So maintenance dose is worked out basing upon the clearance 15 liters into the dose concentration required is only 10 mg per liter, and hence if you multiply 150 mg per hour, it is intravenous infusion. So loading dose is given as a bolus injection once it is administered, but whereas maintenance dose should be given continuously, and hence here it is an intravenous continuous infusion. So maintenance dose dimension is mg per hour. Per hour, this much drug has to be given to sustain the whatever is lost. And hence, like this, you have to calculate the basing upon the normal kinetics and based upon the kinetics in the patient, you can work out these things. And then in the same way, there is another case, and here also involves the calculation of these quantities only, and it is 30 liters per hour and 10. I think the bolus, okay, it's already seen, okay. And knowing these things, you can also calculate the half-life. So in the first case, clearance is 30 liters per hour, volume is 100 liters, and required concentration is 10 microgram. So loading means always the VD into that, and hence the loading dose is worked out to be 1000 mg intravenous bolus injection. And clearance is here 30 liters, and that should be, or again CS is 10 with the result 30 mg per hour, and it is infusion, intravenous infusion. One is the bolus injection, always loading dose are bolus injection only. And if they are also calculated based upon the VD, okay, and based upon the clearance, MD is calculated. In both the cases, the same type of calculation. And further, you can also see the half-life. Half-life is again the same information. If you rearrange, the half-life was found to be 2.3 hours. And steady state should be achieved in 3 to 5 hours. That is general rule, general observation. And hence, in the same manner, this is another case where it can be worked out. Infusion rate always based upon the VD and CSS and K. And here it is in the, it is also a uremic condition, means kidney is got damaged. Here also the dose K is reduced. K means elimination rate constant is given. Instead of clearance, rate constant is given, that got reduced, and hence the dose to be administered also simultaneously got reduced. One observation needed here is in the normal case, the infusion rate is 20 mg per hour, the first part. In the uremic condition, when the kidney function is damaged, the K is reduced to half, and hence infusion rate also got reduced exactly to half, 10 mg per hour. But like that, the loading dose, maintenance dose particularly, is dependent upon the elimination character, and which will be changing in the disease condition, and hence for each patient, carefully analyzing the elimination character, you can make adjustment in the doses, either the dose given or the rate of administration or the dosing interval, etc. And hence this is what in a nutshell about some essential concepts of clinical pharmacy and pharmacokinetics, its application in clinical pharmacy, and hence knowledge of clinical pharmacy, clinical pharmacokinetics is very much essential for clinical pharmacists to perform their duties, responsibilities more appropriately in the true professional manner, and hence overall the subject of pharmacokinetics is very much useful for all research investigators, right from drug discovery development research to formulation research to clinical research. Everywhere the concept of pharmacokinetics plays a vital role, and hence a thorough knowledge and skills in this subject is very much essential. That is the reason why, just to give an overview of the essential concepts only, the topic is unlimited, anything can be discussed. And hence, I thank the organizers, particularly the principal, Deval Garu, and the coordinator, the organizing secretary, and HOD of Pharmacy Practice Department, and the Anupama madam and other coordinators, particularly the management and principal of the Siddhartha College of Pharmacy, for the excellent initiation in these programs during this COVID-19 pandemic period. And anyway, I wish good success to the college and all the teachers. And I know that all the teachers are always busily engaged in teaching and research activity. And this type of activities, earlier, the Alra is very busy in a weekly seminar in one subject or otherwise, the seminar hall. Now, in the same manner, the Dalra may busily be conducting webinars every week, If not continuously, every one day you can fix it up and invite one or other person who is absolutely free like me and continuously deliberate on one or other issue. So wish you all the best to the college and to the principal, HODs and the faculty and students of Siddhartha College of Pharmacy. All the best to everyone.
Thank you, sir. Thank you for your valuable session and valuable information about linear pharmacokinetics and non-linear pharmacokinetics and compartment models and posology. And thank you, sir. Any questions from the participants? Okay, no questions. So thank you very much, sir, for your valuable information and your presence. We're very much thankful to you, sir. Thank you, sir.
Thank you.
Okay, Anupama?
Okay.
Thank you, sir. Okay, sir.
Okay, sir. Thank you all. Good luck.
Thank you very much, sir, for your extensive presentation. Thank you, sir. Thank you for sparing valuable time.
Thank you.
Thank you, principal, sir. Thank you, sir.
Thank you, Anupama.
for. Okay, sir. Thank you, sir.
Okay, right. Okay,
Okay, sir. Thank you. Leave.
All right.
Okay. One by one. Okay.